During our last encounter, we talked about acid-base balance, and it's a complicated issue, which I tried my best to simplify for you. So I want to remind you a few words about the buffers. Okay, we talked about them a little bit, so I'm going to talk about buffers now. So we have three buffers. We have bicarb, we have phosphate, we have protein. Now protein, just think about where you're going to find the most protein. Cells, inside of the cell, because of all the protein synthesis. In plasma, a lot of protein in the solution. Remember, interstitial fluid outside of the cell, is protein poor. We talked about it before when we discussed the tissue perfusion, the capillary perfusion. Okay? Do you follow me so far? Now, proteins have those amino groups that can bind excessive hydrogens, and they have carboxyl groups, okay, that can release hydrogens. So it's all about hydrogens. So you can say, okay, proteins work in plasma, proteins work inside the cells. Bicarbonate buffer. Definitely outside of the cells, in ECF and in plasma. Why? What is the main driving force for bicarbonate buffer? Which gas? CO2. Without CO2 there would be none, right? So since we have CO2, CO2 is produced by cells and is released from the cells into the extracellular fluid and in plasma. It is super important in ECF. Is it present inside of the cells? Yes. Is it super important inside of the cells? Not so much. It's super important in the extracellular fluid and in plasma. Does that make sense? So, how it works? Bicarbonate can bind excessive hydrogens, or carbonic acid can release hydrogens if they're needed. Are we clear? And phosphate is urine, the most important in the urine. Okay? Now, how it works in kidneys. There are, first of all, did we discuss what's more frequent? Alkalosis or acidosis? Acidosis. Now, which two systems can alter, two organ systems can alter blood pH? Respiratory and renal. Renal, right? Cardiovascular, only transport. Renal, okay. So renal and respiratory. Generally speaking, are you normally? Yes. Generally speaking, I don't know of any condition that is associated with, let's say, hyperventilation. You know what I'm trying to say? I cannot think of any long-term pathological condition that forces humans to breathe hard, deep, and fast. Do you understand? I do know of the conditions that are associated with a poor ventilation, say COPD. We clear about it? So when ventilation is poor, what's accumulated in the lungs? CO2. Acidosis. Now, metabolically, and we'll talk about it a little bit more, metabolically, those of you who, a lot of you, I bet the majority of you, you work in the hospitals, or you are associated, or you've heard about it. Metabolic acidosis. Metabolic alkalosis is super rare. Metabolic acidosis is frequent. You know, alcohol intoxication, diabetic complications, kidney failure, you name it. alkalosis is weird. We'll talk about how you can acquire it, but again, it's weird. Okay, so in both cases, acidosis is the most frequent thing. So what do you need to do to get rid of acid in terms of the kidneys? You need to excrete it somehow. You get to put hydrogen ions in the urine. Are we clear? So this here, hydrogen ions in the urine, is our goal. Does that make sense? And when we put hydrogen ions in the urine, it basically automatically means that we need to put bicarbonate in the blood. So that's another way to compensate. Either, reabsorb, quote unquote, reabsorb bicarbonate or excrete hydrogens. Does that make sense? Because bicarbonate, baking soda, essentially bicarbonate is baking soda. It's a base. It can reduce acidity, the blood or extracellular fluid. It can increase pH. So far, am I being clear? So let's focus on reabsorption of bicarbonate. I'm going to tell you one hard, sad truth. Bicarbonate cannot be reabsorbed. Period. There is no reabsorption of bicarbonate. What is reabsorption? It's the transport of chemicals from where to where? Come on. Reabsorption. From filtrate to the blood. That's it, right? So what is that? Remember we talked about the major driving force behind reabsorption. What is it? Gradient of which ion? Sodium, right? So you gotta have sodium-potassium pump here. There it is. It's there. And you gotta have some kind of transporter, secondary active transport, on the apical side of the tubular epithelium. So far, do you follow me? There's only one little trouble if you look at this picture. There's sodium-potassium pump, of course. But there is nothing that would distantly remind you about bicarbonate transporter. There's none. The mechanism of bicarbonate, I put it in the quotation marks, reabsorption, is incredibly elegant. So here's what happens in the tubular epithelial cells. Look here. First, foremost step. CO2 combines with water. You see on the bottom? CO2 combines with water. That combination leads to, as usual, carbonic acid, which then dissociates to the bicarbonate and hydrogen. And here comes the most fascinating moment. Bicarbonate that is formed inside of the tubular cell is transported into the blood by the basolateral membrane. Okay? Hydrogen is excreted into the lumen of the tubule, into the filtrate, and in the filtrate hydrogen recombines with bicarbonate, which is normally present there. This bicarbonate and hydrogen form carbonic acid. Carbonic acid dissociates in water and CO2. CO2 diffuses back in the epithelium, interacts with water, produces another carbonic acid, which releases another molecule of bicarbonate. which is transported in the blood. Hydrogen is released here, binds to bicarbonate. Does that make sense? So that bicarbonate is initially filtered out, so it's a product of filtration. But the elegance of this mechanism is that essentially, if you look at this and you sort of put on the side burner this process, essentially what happens is hydrogen ion is excreted and bicarbonate is in the blood. Does that make sense? But this is done sort of indirectly. There's no direct reabsorption of bicarbonate. There is no direct secretion of hydrogen ions. Does that make sense? Now, what drives hydrogen secretion? That's right here. Sodium diffuses into the cell, providing energy for hydrogen excretion. So it's anti-porter. Does that make sense? I think it's just super elegant, really. So you have carbon dioxide that combines with water in the cell. Carbonic acid that is formed is dissociated into bicarbonate and hydrogen. Bicarbonate is taken into the blood. That's your bicarbonate. Is it acid or base? Now, here's the deal. I want to make this comment. Listen up. If you bring base into the blood, it's basically the same as if you excrete hydrogen. Does it make sense? It's like a double-edged sword. You can either add some base or throw away some acid. So in this case, you add the base. Does that make sense? Okay? So if there is a lot of CO2 in the blood, there will be a lot of this formation in the tubular cell. There will be a lot of bicarbonate produced, and it will decrease the acidity of the blood right away. Because the rate of bicarbonate formation in the lumen will go up. Does that make sense? Because we'll get to different types of acidosis, but basically this is the elevated CO2 is acidosis in the blood. Does that make sense? Okay? Now, two types of acid-base abnormalities that we discussed are renal and respiratory. True? If there is a respiratory acidosis. Let me use acidosis because it's more frequent, but same is applied to alkalosis. If you have a patient with respiratory acidosis, can respiratory acidosis be fixed by the by the respiratory system? No. It says right there, respiratory system cannot compensate for respiratory acidosis. I never tell you why. It's a stupid question, really. Yeah, because respiratory system is compromised. Respiratory acidosis develops because you cannot breathe normally. How can you compensate for it? For something that is caused by your breathing if you cannot breathe normally. Does that make sense? Same goes for metabolic. What are the causes of metabolic acidosis? Most likely. Well, a lot. Renal failure. Okay. Can it be something else? Yes. Well, basically, diabetic ketoacidosis isn't necessarily because of renal failure, but kidneys are overwhelmed. They just cannot process that much. That's the problem. Okay? So, metabolic acidosis cannot be compensated by kidneys. Respiratory cannot be compensated by lungs. Are we clear? These systems will try to work for each other. Does that make sense? So, I'm going to walk you through these tables and tell you what each table means. So first, this table here summarizes the symptoms and signs and mechanisms of respiratory and metabolic acidosis and alkalosis. Respiratory acidosis. Decreased ventilation or gas exchange. If ventilation is decreased, it's most likely what? If you cannot breathe normally, what is compromised? Respiratory passages. Obstructive or restrictive disease? Which one? Obstructive, right? Because your respiratory passages are obstructed. Gas exchange in the lungs is compromised. That's most likely restrictive. Emphysema, different types of alveolar damage. Does that make sense? Can it be both? Sure thing. Are we clear about it? So, insufficient ventilation or insufficient gas exchange means CO2 accumulates in the blood. Can you imagine this condition be chronic? Absolutely. Respiratory alkalosis. Hyperventilation. I have pretty rich imagination. I can imagine this to be chronic, but I've never heard about one. I've never heard about any patient that would chronically hyperventilate. So it is usually temporary. It's not of that much concern for us. Does that make sense? And you know what to do. Just give them a bag. Metabolic. Metabolic acidosis. Now, this is more interesting. In metabolic acidosis, which ion, don't look there, look at me, which ion is going to accumulate in the blood? Which ion is acid, quote unquote? Hydrogen ion. So hydrogen ion will it really accumulate in the blood? Why? What prevents accumulation of hydrogen in the blood? Say again? Bicarbonate buffer. Very good. Bicarbonate buffer will prevent. But we will use bicarbonate to sequester hydrogen from the blood. Does that make sense? Which means bicarbonate levels in the blood will go down. Are we clear? Diabetes, alcohol consumption, or diarrhea, chronic diarrhea. When essentially we excrete bicarbonate in feces. We're clear. So all these conditions I can quite realistically imagine. Diabetic ketoacidosis, alcohol intoxication, for sure, chronic diarrhea. Metabolic alkalosis, much less frequent. Vomiting, profuse, violent, long-term vomiting. When you vomit the content of your stomach, what happens to pH in the stomach? When you get rid of all that acid? Initially, pH goes up, which is compensated by parietal cells that will produce more acid. Where they take this acid from? Well, acid, I mean CO2. Blood. So essentially, acid will be, quote-unquote, taken from the blood and shuffled into the stomach. And if you keep vomiting it, you essentially vomit out acid that you normally have. Does that make sense? It's not really frequent, that much of a vomiting. So usually respiratory alkalosis is transient. Now, what are the general symptoms? For acidosis, coma, low pH, depresses neuronal activity and leads to neuronal inhibition in coma. High pH leads to excitation of nervous system. So quite an opposite. Okay, muscle overactivation, seizures, and it may actually lead to the respiratory arrest due to the tetanic paralysis of diaphragm. Little digression about alkalosis. There's a textbook that is used in many allied health schools and medical schools called the Textbook of Medical Physiology, authored by Guyton and Hall. It's a classic textbook. And in the chapter on nervous system, when they talk about, pH influence over the activity of neurons, they have a wonderful passage. I don't remember it verbatim, but they say, and high pH overactivate nervous system. For instance, if you will ask a person with epilepsy to hyperventilate, you will cause an epileptic bout. Which I think it's a pretty evil idea to put in the textbook, but essentially it can happen. And speaking of which, one of the treatments for epilepsy is ketogenic diet. Because when somebody is in ketosis, the person experiences consistently lower, slightly lower pH. It's still in the normal range, but it's lower. It decreases excitability of neurons in the CNS. And it was shown to reduce the frequency of epileptic bouts. Now, let's recall the responses to respiratory and metabolic changes. When respiratory system fails, which system will try to compensate? Renal. So, how renal system will respond to acidosis? It will try to excrete hydrogen ions. Very good. Go back. It's kind of a huge mess over here. But let's take a glance. With every hydrogen ion excreted, what goes back into the blood? Bicarbonate. So the result of renal response, and this is what I have in this table. So this is the response, right? And this is the result of this response. What you're going to see in a patient with respiratory acidosis is higher levels of bicarbonate in the blood. Does it make sense? So these higher levels of bicarbonate in the blood, they come from the renal response. This is the symptom of the respiratory system dysfunction. Do I make sense? Do you understand those two things? So high CO2 in the blood is because lungs don't work. High bicarbonate is because kidneys do work. Do I make sense here? And the same response you will see in alkalosis. So what is the response? Try to keep hydrogen in, which means lower levels of bicarbonate in the blood. That's exactly what you're going to see. Bicarbonate levels in the blood will start to climb down. Okay? I'm sorry. But And concentration of CO2 will be down in the very beginning as well. Does that make sense? Now, much more frequently, you're going to encounter metabolic acidosis. Let's say due to the renal failure. So metabolic acidosis will manifest as the higher acidity, right? Now, Lexi mentioned what's going to happen to all those hydrogen ions that accumulate in the blood. They're going to bind what? Hydrogen ions in the blood. What are they going to bind? Bicarbonate. Okay. That's bicarbonate. Bicarbonate. They're going to bind bicarbonate. Which means in acidosis, bicarbonate concentration in the very beginning will be low. Is that understood? People with metabolic acidosis, what pattern of respiration do they have? How do they breathe? High ventilation rate. Does that make sense? Do I understand why? They try to get rid of CO2. They try to get rid of CO2 to increase pH, to reduce acidity. So, how this response is going to change concentration of CO2 in the blood? It's going to go down. Does that make sense? You see? So, initial symptom of metabolic acidosis is low bicarbonate. After the response, you're going to see low CO2 because they ventilate really hard. Does that make sense? Okay? In metabolic alkalosis, exactly opposite. You see elevated levels of bicarbonate in the very beginning. That's the definition of alkalosis. The response, decreased breathing rate, which elevates partial pressure of CO2 in the blood in an attempt to compensate for this effect. Does that make sense? So, I really want to kind of remind you that this is what it's, this is what it starts with. These are results of responses, okay? This is start. These are responses. Are we clear about it? Now, that actually concludes the kidney physiology. I want to mention one thing, since it's not in PowerPoint. I'm really strict about it for myself. I'm not going to ask this question, but kidney stones. You've heard about them, right? Mostly calcium accumulation, different mineral accumulation. How do we treat them? Do you know? Ultrasound lithotripsy. Basically, we shock kidney stones. They become smaller, and then they are excreted with urine. As far as I heard, it's painful, really, really painful to excrete them. And as far as I've heard about kidney stone itself, it's incredibly painful to have them. Often it is considered to be the worst pain a person can experience. My former science advisor that I did PhD with, he had kidney stone when he was working in England. So basically, like, very basic medical insurance. But he was working in a really nice half hospital facility, so he ended up to being injected with morphine. It was half legal. I mean, it was a doc who injected, but it was sort of off the charts. And yeah, it took care of the pain, but he said it was last time I tried opioids. Because he realized second time, and he's going to be hooked. So he said it was like best night of his life. There's no pain, and he just felt great because of the drug. But that's how bad. So regular painkillers would not take care of this. Now, this is fairly basic anatomy. I will focus on what I really want you to know about these structures. Urine, as it flows out of the kidney, ends up in the ureter. Okay. Ureters are long and muscular. Unless there's urine in them, they collapsed. You can see they practically collapsed. They're stretchable. The layer of smooth muscle helps to propel the urine towards the urinary bladder. A few things that I want you to know. There's no submucosa. You have epithelium, you have mucosa, and you have adventitia. Okay? I want you to know the type of epithelium there. It's transitional, which is sort of unique for urinary system. And then there's urinary bladder. It's pretty thick, really thick layer of smooth muscle, which allows to contract and expel the urine. Does that make sense? You understand that? It's basically a muscular sac. Mucosa is again transitional epithelium. There's no submucosa. Smooth muscle, the detrusor muscle, is pretty strong. 3 layers, that's a lot. Normally the volume is about ½ liter. It can stretch, can hold up to 1 liter of urine, which is a lot. Can it burst? Yes. That was actually a punishment in ancient Rome when convicted, because usually it was performed on males because it was easier. They put a knot on penis, blocking the urethra, and inserted a funnel and started to fill them up with wine. So it was like 2 gallons basically. Not immediately, because otherwise they'll just totally explode, but more slowly. So eventually the urinary bladder burst, and it was pretty painful death in the end. And of course they had no surgery back then, so like now urinary bladder is this like a critical organ for life. No, you can put artificial back basically there. It's possible. Okay, so you can live without urinary bladder. But back then there was no option like that. So, interesting thing about ureters is that here, these are the points where ureters enter the urinary bladder. And when urinary bladder fills up, ends of ureters, they close to prevent the backflow. And I will tell you the reason for that. So from the urinary bladder, urine flows through the urethra. That's the female urethra, which is pretty short, as you can see. Okay. Did I say male or female? Good. So that's female. Yeah, female is short. Male is longer. Okay, and in males, urethra and basically ejaculatory duct are at some point the same. So that's kind of slight anatomical difference here. Okay, in females, The vaginal orifice and urethral orifice are totally different. In males, penile orifice and urethral orifice are essentially the same. Okay? So the mucosa, epithelium, is transitional closer to the bladder, and it turns into the stratified squamous, basically around the orifice, which makes perfect sense because there's some rubbing can happen. Okay? Female urethra is much shorter. There's no actual separation by parts. It is, though, the outflow of the urine is controlled by two sphincters, external and internal urinary sphincters. Now, external urinary sphincter is controlled by somatic nervous system. Does that make sense? So you consciously control external urinary sphincter. I'm talking consciously. Internal is controlled, is stimulated by parasympathetic. Parasympathetic signaling relaxes it. Are we clear? How well can we control external urinary sphincter? Fantastically well. There was a pretty inhumane experiment. There were a lot of them in the 70s and partially in 80s. That experiment, though, was done with the consent of participants, a bunch of students. I think it was in some institution in California. It's like a university. So what scientists wanted to look at is the strength of social prohibitions. Socially, it's not really acceptable to pee in your pants. Right? For adults. So a group of students were locked up in the room with basically chaperones in there, and chaperones did not let students to pee like in the corners of the room. So, and students were given plenty of drinks, basically forced to drink. The idea was to see how soon students will start to pee their pants, how strong those social prohibitions are. They were exceptionally strong. The majority of students passed out, but did not pee the pants. I mean, when they passed out, they did, but the level of stigma associated with peeing pants was so strong in adults that they actually held the urine for that entire time. And the urge was so strong that knocked them unconscious. Okay, nobody got hurt. It's fine, but that was pretty interesting. So we control that external urinary sphincter really well. Now, interesting difference between the male and female urethra. Female is much shorter, right? And that determines why level of urinary tract infections in females is much higher. Urethral orifice is close to anal orifice. There's a cross-contamination possible. And can it happen in males? Absolutely. The only problem is that in males, bacteria have a longer way to climb. So they can get stuck in the urethra, and males have 3 parts of urethra, 3 zones. They have prostatic. Guess where? That runs through the prostate gland. Very short membranous urethra, okay, that goes basically through the external urinary sphincter. And then they have spongy urethra. That's the part that goes through the body of the penis. So in males, urinary tract infection, bacteria have much longer path to get through to end up, say, in the urinary bladder. Infection of the urinary bladder is called cystitis. Okay? Now, I told you before that when urinary bladder is full, ends of ureters are closing. The main purpose of this response is if there is any bug in the urinary bladder, closed ends of ureters prevent this bug from getting through urethra to kidneys. Does that make sense? Actually, this region right here that is bounded by the orifices of ureters and the urethra, it's called trigone. That's the region that is infected most frequently because it's just next to the urethra, okay? And they really don't have a lot of opportunities to get to other parts of the urinary bladder. Interestingly enough, that people who have to hold their urine for extended period of time, they are more prone to urinary tract infections and to kidney infections. Because basically, think about this. Every time you pee, simple flow of urine removes microorganisms. If you hold it, there's better chance for them to migrate towards the sterile parts, the urinary tract, including kidneys actually, lead the infection. And there's a profession in which people hold the urine for quite a long period of time. Truck drivers. They try to stop as little as possible, and they often, you know, when you drive a car, you usually have a cup of coffee and a cup of something, you know, with caffeine next to you. Okay? Caffeine is diuretic. So that's going to help urine to form, and you will try to keep it in for as long as possible to cover as many miles as you can before you stop. So sort of a professional disease of people who sit for an extended period of time. Does that make sense? Now, what is the reflex for urination? It's all about stretch. When urinary bladder feels, the bladder wall stretches and stimulates mechanoreceptors. Mechanoreceptors send a signal to the brain. You do appreciate the feeling of the full bladder. You know that, right? You want to pee. And to the pontine centers. Okay? So micturition center. Now, these two centers, one of them stimulates urination, one of them inhibits urination. So let's say you are an adult and you are on the bus and you have an urge to pee. Am I going to pee? Probably not. Okay? So your cortex will stimulate the storage center, which will inhibit parasympathetic system and activate sympathetic and somatic activity. And essentially, it's going to keep the external urinary, external urethral or urinary sphincter closed. You still have an urge, but sphincters are closed. Does that make sense? Now, when you're ready to pee, then cortex stimulates micturition center, which inhibits sympathetic and somatic activity, okay, and increases parasympathetic activity. So I want it to be very specific. First, it's the concerted effort. It's like a lot of things have to start moving. When you pee, internal urethral sphincter has to open. This is parasympathetic stimulation. Smooth muscles of the bladder have to contract. This is parasympathetic stimulation. Does that make sense? In this case, sympathetic and parasympathetic systems, they play teeter-totter. If parasympathetic activity is up, bladder contracts, sphincters open. If sympathetic activity is up, bladder does not contract, it is inhibited, and sphincters closed. Are we clear about it? So sympathetic activity promotes urination, sympathetic inhibits it. As you can see, your bladder and internal urethral sphincter are under autonomic control. You have no power. Does that make sense? However, your external urethral sphincter is under somatic control. You have a power. Does that make sense? So you consciously open the sphincter to let the urine flow out. I'm sorry. Does that make sense? Here's the deal. What happens in kids? There's no storage. That signaling isn't developed yet. Does that make sense? And none of this. So, I mean, they have all the wiring. They just don't know how to do it. Does that make sense? So infants truly have a reflexive urination. When they feel it, they pee. Does that make sense? No reflex. And again, systems. I'm going to ask you about which system controls which sphincter. Somatic, external. Parasympathetic activation, internal. I'm going to ask you where the centers are. Remember, they are in pons. That make sense? Did I make myself clear? Any questions? I'm going to introduce you to the anatomy of the male reproductive system. It's quite fascinating. It's actually pretty elegant, and again, a lot of pipes. Two main parts of the male reproductive system: penis and scrotum. Okay, so we're going to start with the scrotum, which contains testes. So this is, we actually in the department, you know, like if you have a test, test is 50 questions. But if we have like a small test, small tests in the departments, in our department, we used to be called testes. 25 question test is testes. Each testicle, yes, is surrounded by tunica vaginalis. It's actually an extension of visceral peritoneum. And you can see on top of the testicle here and here the structure that is called epididymis. Epididymis is a very finely folded tube, okay, where sperm matures. Epididymis extends into the vas deferens, which leads sperm to the penis. Now, scrotum on the outside, of course, it's a skin sac, and subcutaneously you can see a fascia which contains muscle called dartus muscle. Under another layer of connective tissue, there is a muscle called cremaster muscle. Can you see it here? So cremaster muscle allows to regulate scrotal temperature, which is absolutely critical for the maturation of sperm. When it's cold outside, testes are pulled in to maintain the ideal temperature for sperm maturation, about 35 degrees Celsius. When it's hot, testes are pulled out, okay? They descend to cool down and to maintain that same temperature. And it was actually shown that overexposure of scrotum to higher temperatures, either by, so it was shown, for instance, that in males, if a male takes a really, really hot bath, extended bath, the survivability and fertility of sperm significantly decreases. It is not killed, I mean, unless you boil them, it's not going to work. But it decreases the fertility of sperm. Okay? It actually can be very clearly seen in rodents. Those of you who have little kids or not so little kids, they often want small pet. And a fairly worry-free pet is a hamster or mouse. Mice, male mice, have, as far as I know, the highest ratio of the testicular size to the size of the body. And if it's hot, you can see how testes descend from the mouse body. You can actually see that same. We had hamsters. You know, if it's hot, oh, you can notice that's male right away. Okay. If it's cold, you're going to pull it back. Now, in some cases, in some males, testes do not descend in childhood. When boys are being checked by family doctor, by physician, as they grow up, one of the part of examination is to check testes actually descend, if they are in the scrotum. Because if they aren't, the condition is known as cryptorchidism. And it's can be fixed surgically, okay? But the problem is if it's not diagnosed timely, it leads to infertility. It's untreatable infertility. Because cryptorchidism means that testes are constantly inside of the body, and they are constantly exposed to high temperatures, and spermatogonia do not develop properly. Sperm does not mature properly. And that may lead to serious problems later in life, mainly associated with psychological problems. We have a friend who was not timely diagnosed with that condition, and it's when he discovered it was really a shocking thing for him. So he cannot have kids, basically. And he discovered it when he was like in late 30s. He didn't know that that happened to him. Yeah, so that's pretty sad. Now, so the dartus muscle and cremaster muscle, they used to pull testes in and descend them out. All the nerves and blood vessels are encased in what is known as spermatic cord. There's a condition which is, it's not a hernia, I blanked on the name. Basically, in some cases, especially among weightlifters, spermatic cord can become twisted because of abdominal muscles. It can kind of turn it, and if it becomes twisted, there's a strong pain around the spermatic cord. It again can be surgically treated, but it's rather unpleasant, and it shuts down the blood supply and nerve supply to testes, so that may affect the sperm maturation, so it has to be treated timely. When sperm is produced, it leaves epididymis and goes through the vas deferens and ejaculatory duct, eventually in the urethra, as you can see. Prostate urethra, membranous urethra into spongy urethra. And then when sperm is ejaculated, it's ejaculated from the urethral orifice. The sperm passes through, it experiences the influence of several glands, which functions we can talk about. The seminal gland, the prostate gland, and the bulbourethral gland. The space between the penis, the coccyx, the anus, and the ischial tuberosity, this triangle, is called perineum. Okay. Now, anatomically, if we talk about penis, we have a root of the penis, and we have a shaft of the penis. Here, this part is called glans penis, the most distal part, and glans penis. Right here, it's a cross section. Remember, it's a cross section. So glans penis is covered by prepuce or foreskin. Okay. So foreskin can be removed in the process of circumcision. Now it becomes a controversial issue because it is done on children without their consent. About 60%, whatever number, 60% of newborns in the United States are circumcised, either for religious or medical reasons. Medical, I mean hygienic reasons. So I'm going to put ethical questions aside. I'm not going to discuss whether it's ethical or not. Obviously, too much. I don't want to touch it with a 6-foot pole. From the medical standpoint, why in Jewish and Muslim culture circumcision is mandatory? It's mostly, as we think now, due to the climate that those ethnicities, those folks lived in. It's hot. Water is scarcely available, so personal hygiene is often not possible. Removal of foreskin decreases the rate of infections associated, basically sexually transmitted or urinary tract infections in boys. Okay, so it was warranted from the hygienic standpoint. If a child can maintain glans penis clean, okay, if the child can maintain his own personal hygiene, circumcision is technically not medically relevant. It was shown pretty consistently that it reduces not only HIV, risk for HIV transmission by about 60%, but also reduces the risk for the transmission of other sexually transmitted diseases. Again, if you think about it, there is a method to reduce the risk for HIV transmission or any other STD transmission by 99%. There's a condom. Okay? So, if you can be sure that the child will be compliant with a safe sex throughout their life, then circumcision is not medically warranted. In some cases, circumcision is performed in adults in case of condition known as phimosis. It's inflammation of the foreskin. That inflammation causes fibrosis and tightening of the foreskin, which prevents, actually causes really painful erection. Foreskin is basically very tight around the penis. So in this case, circumcision is performed. Now circumcision is an outpatient procedure. So it's like one-day deal. You go in, procedure's done, you go out. Okay? So, what's inside? I apologize if I'm going to hurt somebody's feelings, but there are some misconceptions that are kind of joking misconceptions and funny misconceptions and childish. It's not a bone, although some people think it is. in the kids. It's not a muscle. There's no way you can train it. It's a special kind of connective tissue labyrinth, should I say? You know what I'm saying? It's like it's a sponge. Okay? Sponge with really hard and resilient, made of mechanically resilient material. And the names of the parts actually suggest that. So you have corpora cavernosa, from Latin it basically is cavernous body, and corpus spongiosum, spongy body. So you have three structures here, okay? These are blood vessels, and the urethra are inside of corpus spongiosum. So when there is an erection, those spongy bodies are filled with blood. When they're filled with blood, penis engorges, and there is an erection. We're going to talk about erectile dysfunction, but one of the main mechanisms of erectile dysfunction in males is, of course, poor blood flow. Sometimes there are psychological issues, but essentially those psychological issues, they prevent proper vasodilation in penis and prevent the elevated blood flow to the penis. Okay? So I wanted to finish the, let's talk about this system of pipes, and we'll take a break. So, sperm. Sperm is produced in testis, as I mentioned. So if you would look at the cross-section, the testis, you will see that there are a lot of little tubules called seminiferous tubules. You see those really bent. The idea here is that if you have those tubules bent, you can package a lot of them. Does that make sense? And since that's where sperm matures, produced and eventually matures, you can produce a lot of sperm cells. It's just a lot of tubular lumen to have. Okay? So as sperm is produced in seminiferous tubules, although being not really functional and mature, it is released through the straight tubules, very, very short trunks here, into the network known as reti testis. From reti testis, sperm enters, here you go, epididymis. You see those heavily folded tubules here? The total length here in the male, in human male, can be about 6 meters. Now, my own encounter with epididymis happened in mice. I did mouse surgery. We isolated testicles, actually one testicle, mouse. And we opened it up to flatten out cremaster muscle, because it was great to look at the blood vessels in it. And when you open up the cremaster muscle, essentially have a testicle. It's a free one. One thing that I learned pretty fast is that you must not damage epididymis. It's covered with a thin membrane. If you let it out, this network of thin tubules is everywhere. It covers the microscopic field of view. You cannot see anything, and you cannot put it back. One of the research associates that worked with mice, she did a brute force thing. Since it was a terminal surgery, she actually, after the cremaster muscle was isolated, she tied the testis down to block the blood flow. And eventually, the blood vessels that went to testis through the spermatic cord, they thrombosed, to just cut it off. Because it's inconvenient to have it in the field of view. Okay? So it's super long. Epididymis extends into the ductus or vas deferens. So when sperm is produced in the seminiferous tubules, it enters epididymis and slowly moves through and matures and matures and matures. And the mature sperm during the ejaculation is ejected from epididymis through the vas deferens, through the ejaculatory duct into the urethra. It is functional for an extended period of time. And actually, vasectomy is, before it was basically cutting vas deferens, this is vas deferens, so ductus deferens, which was irreversible but the most efficient way to of the male contraception. Obviously, if you cut it, you cannot do anything about it. Now, surgeon can tie it, so there is no passage of sperm, right? But in case man rethinks and decides that he wants kids again, this ligation can be removed, and technically it's intact, and the male can produce the functional sperm again. Does that make sense? Let's take a break. We'll talk about accessory glands, and we'll talk about mechanisms of ejaculation and erection later. Study guide.